Hello, and welcome to Lecture 6.5, The Human Microbiota. In this lecture, I will tell you about the microbes that live on you and in you, but more importantly, I will tell you some important things to think about when you think about them, and some important things they do, where to find them, and a few ways they go wrong. So let's get into it. I'll start in this video by talking about just what it is and why we should care about it. So the microbiota is the microbes that live on us and in us for a long time. And so what you see on the screen, normal and resident, means they live on us for many generations. So you might hear someone say the normal microbiota, the normal flora, the resident microbiota. That all means the same thing. It means organisms that live on us or in us for many generations. Think about doubling time. You may want to pause the video and go back and look up doubling time. But if an organism can double in 20 minutes or even an hour, how many times does it double in a day? How many times does it double in a year? When you think of it that way, you realize that these microbes that live on us and in us for a lot of our lives, we find the same species on us and in us, we are their universe, and they are in us for countless generations. They are evolving in us and mutating in us. So they are their residents. In some terminology, you'll see microbiome and microbiota. These two words are sometimes used interchangeably, but they do have subtly different meanings. So the microbiota is the species and the strains, the genera, these cells in us. The microbiome is their genes, their DNA. And we make that distinction when we think about the capabilities they give us. So when we think about what those cells could do for us, one window we have into that is their DNA and their genes. And when we look at them through that window, what we see is that they have enormous potential to do biochemical reactions with their enzymes, enormous potential to change the chemistry of our gut in particular, but to help us do chemical reactions we can't do using our own cells. So microbiome and microbiota, people will know what you mean. Just know microbiota is the term for the cells and the species. Microbiome is the term for the DNA and what those cells can do, if you want to be a stickler. Flora is probably the term you're going to hear most often if you're going to work in clinics. And I don't like it because it means plants. And bacteria, archaea, fungi, they are not plants. If you know what plants are, you're not going to confuse them with bacteria. So I don't use the word flora, but understand lots of people do, and they use it to mean microbiota. Okay, so you may have heard that your microbiota outnumber you. And you might want to pause the video and go back to scientific notation and powers of 10. Because we're going to use some of those. What you may hear is that there are more bacterial cells in you than human cells, and that is probably true because you have something like you have a few kilograms of fecal material in your colon, and half of that is bacterial by mass. Well, the mass of any one bacterium is very, very small. If you have a kilogram of them, you have an astronomically huge number of cells. You already had that astronomically huge number of cells. You have something like 30 trillion cells. Well, you may have 100 trillion bacteria. It doesn't really matter. What matters more than that is what enzymatic reactions can those bacteria do that your body cannot do without them. And we will get into that a little bit. So let's just go through. Oh, right. And the other thing, for every bacterial cell on Earth, there are 10 virions of bacteriophage on average, which means that you are way outnumbered by bacteriophage, but they don't affect you. The only way they affect you is by killing off groups of bacteria in your everywhere bacteria are found. And where you find the microbiota, generally speaking, is on external surfaces. And so skin is an obvious one, but also the lumen of hollow organs that have openings to the outside. Those could be considered external. So like the epithelia inside your nasal cavity and your mouth epithelia, you find lots of microorganisms there. They're part of your microbiota. The colon, so not the whole digestive tract, not the stomach, not the small intestine, but the colon, you'll find lots and lots of bacteria. And the vaginal mucus is where you would find lots of microbes. So we'll come back to that. But what all those things have in common is they are external surfaces. They are not your connective tissues. They're not muscle. They're nothing like that. If we ever see microbes in our connective tissues, we immediately mount an immune response to get rid of them. You will read and hear interesting things about the microbiota, and you may have questions that I certainly can't answer, and science has not yet answered, and that is because of how complex the microbiota are. This comes from the fact that individual strains might be different from each other in how they affect us. Well, if that's true, we should want to know all the different strains that are in us and on us. And you might want to pause the video and look up the taxonomic levels: genus, species, order, class, domain, strain. What are all those? Strain is the smallest level. Two different strains of a species may differ by one mutation. That's all it takes to make a new strain. If you can tell the difference between them in some way, they are different strains. But more often than that, it is two different strains would have different enzymes, a few different genes, and those different enzymes might affect us. So the challenge is that we probably have 500 to 1,000 species in our colons. And who knows how many individual strains that is. And more than that, we can't see those strains unless we sequence the genome of every bacterial cell in our colon. Our scientific techniques for figuring out what's there don't tell us what strain they are. They tell us what family they are, what genus they are, and sometimes what species, but that's it. So we can't even see the complexity. Yeah. Okay. Well, that's enough for what they are and why we should care. In the next video, we will look at their development of the microbiota.